Cardiac muscle, as we said, is striated in the same manner as skeletal muscle cells, but it differs in the sense that it contains only smaller cells, number one, that are highly branched. There is typically one, occasionally two, centrally placed nuclei, which is, again, a diagnostic feature of them histologically. But the key feature that is an easy way to identify a cardiac muscle array is the intercalated disc. The intercalated discs appear as an array of junctional specializations that are visible as an intercalated disc at the light microscopic level. But at the EM level, we now know that such discs contain a continuing array of gap junctions and adhering junctions. The gap junctions basically allow the spread of the electrical conduction through adjacent cardiac muscle cells, whereas the adhering junctions help maintain the structural integrity of the contracting muscle fibers. Note that in the next picture, which is a histologic view, we see an array of individual striated cardiac muscle cells, and you can see very distinctly that the nucleus is centrally placed and the arrow is pointing to the intercalated disc. At the electron microscopic level, we see the structural features of an individual disc. Note that end to end, the adjacent cardiac muscle cells are joined by a specialized desmosome that's known as a fascia adherens, where intermediate filaments are linked to the adjacent cell membranes. At right angles to that, at the tip of the arrow, is an electron-dense gap junction. Note again that it does not have any association with the cytoskeleton of the adjacent cells, and is there simply to spread a wave of depolarization between the highly branched cardiac myocytes.